Hello students. So students, today we are going to perform the third practical of your biology syllabus. And in today's experiment, we are going to perform certain biochemical tests. Now let us see what will be the aim of this experiment. The aim of this experiment is to test the presence of sugar, starch and proteins from the suitable plant and animal material. And the first test that we are going to perform is test for sugars. Now students, there are certain requirements to perform this test. We need some sugarcane juice, distilled water, Benedict solution, burner, test tube and test tube stand. And the principle for this test is that all the reducing sugars like glucose, fructose, lactose or pentose under alkaline condition will reduce cupric ions to cuprous ions. And the cuprous hydroxide that is formed on heating will turn into cuprous oxide. Now let us also see the procedure for this experiment. To perform this test, we require some sugarcane juice in a test tube. In that we have to add distilled water and the Benedict's reagent. So Benedict's reagent is the one that is used to determine sugars. And after adding the Benedict's reagent, we have to heat the test tube for about 2 minutes and observe the color change. Now students, let us first see how this test is performed and then we'll discuss the observation and the result. So students, let us see the working process for the first test, that is test for sugars, where we will see the presence of glucose. Now for this experiment, we need a test tube. In a test tube, we will take some sugarcane juice. Now this is a diluted sugarcane juice. Okay, and in this we will add about 2 to 3 drops of Benedict's reagent. So Benedict's reagent is the one that is used for the detection of glucose. And when we mix it, you will see that the initial color is blue. Okay, now we will heat it. And on heating, if the color changes to yellowish orange coloration, that means glucose is present in it. Okay, so let us see the heating part and then we will see the observation. So students, as you can see, on addition of Benedict's reagent into the sugarcane juice, the initial color that we get is blue coloration. Okay, now what do we have to do? We have to heat it for about 2 minutes. And as we heat it, we will see that the color slowly changes to yellowish orange coloration. Okay, so if the color changes to yellow or orange coloration, that means glucose is present. So now we will just heat it. And when you are heating it, you have to make sure that you give a time interval in between. If you heat it continuously, all the content in the tube will just pop out. Okay, so you don't have to heat it too much, just give it some interval in between. Yes, you can already see that the color is changing to orange coloration. We'll just switch off the flame. Yes, can you see the color has changed to orange coloration? This means that glucose is present into this sugarcane juice. Okay, now as you have seen the working part for this test, we had taken a test tube in which we added diluted sugarcane juice and then the Benedict's reagent. The initial color that we obtained after adding the Benedict's reagent was bluish green coloration. And on heating it for about 2 minutes, the color changed to yellowish orange color. Okay, so as you can see in the picture, there was a formation of yellowish orange precipitate in the test tube. And on the basis of this, we can determine the result as that the sugar was present in the given sample. Now let us see the second test. The second test is test for starch. And the requirement for this test is some potato pieces and Lugols iodine solution. The principle for this test is that the starch, it reacts with the iodine solution due to the formation of intermolecular charge transfer complex and this turns the color of the starch into an intense bluish black coloration. The procedure for this test is we have to take certain pieces of potato on a slide and treat it with iodine solution. So students, iodine solution is the one to determine starch. Now let us see how it is performed, and then we'll discuss about the observation and the result. The second test that we are going to perform is test for starch. So we are seeing the presence of starch in potato. Now for this, it is a very simple experiment. We just have to take a piece of potato, just a slice of potato, place it on a watch glass, and we have to add Lugols iodine solution to it. Now if on adding the Lugols iodine solution, the color changes to bluish black coloration, that means starch is present. So we'll just add about 2 drops of Lugols iodine solution to it. And if you just keep it for some times, you will see that the color changes to bluish black coloration. I'll attach this picture into the next slide where you can see the color change very clearly. Okay, so as you have seen the simple process for this test, we had taken some potato pieces on a watch glass, and on that we had added the iodine solution. And the observation was that the starch grain, it appeared blue on staining with iodine solution, as you can see over here in the picture. And on the basis of this, we can determine the result as that the starch is present in the given sample. Now after this, let us see the third test. The third test is test for proteins. And the requirement for this test is some test tube stand, test tubes, test tube holder, egg albumin sample, 20% of NaOH, that is sodium hydroxide, and 1% of copper sulfate solution. The principle for this test is the proteins are made up of amino acids, as we know, and these amino acids are joined by the peptide bonds. Now the peptide bonds, they form the complex with the reagent and they give colored product. Okay, and the procedure for this test is we have to take about 2 ml of egg albumin in a test tube, and we have to add equal volume of sodium hydroxide to it, and then mix it thoroughly. After mixing it very well, we have to add 2 drops of copper sulfate, and then we again have to mix it very well, and then there is a color change that is seen. So let us see the working part, and then we'll discuss about the observation. Now we'll do the third test. The third test is the test for proteins. We have to detect protein into the egg albumin sample. Now for this test, we require the egg albumin sample. We require copper sulfate, CuSO4, and we require NaOH, that is sodium hydroxide. Now how do you do this experiment? In a clean test tube, the first thing that you take is the albumin sample. So about 5 ml of albumin sample, and we have to take similar amount or same volume of NaOH. So I'll add equal volume of NaOH to it, and we have to mix it well. Okay. Now after mixing it, the next thing that we have to add is 2 to 3 drops of copper sulfate. And again we have to mix it well. Now after mixing it, the color will change to violet coloration. So if the color changes to violet, that means proteins is present into this albumin sample. So let us just shake it and see the color change. Yes. You can see a beautiful violet coloration into the test tube. This indicates that protein is present into the albumin sample. Okay, so as you have seen how it is performed, in the test we had taken some egg albumin sample in which we added NaOH and CuSO4, and then we mixed it thoroughly. After mixing it, the observation was violet coloration that appeared in the solution. And on the basis of this, we can determine the result as that the proteins are present in the given sample. So students, this was all about this experiment where we performed 3 tests, that is test for sugars, test for starch and test for proteins. In the next experiment, we are going to analyze the urine sample for certain constituents.